talk about excitation, contraction, coupling, basically putting all of what we said together and thinking about how we can regulate muscle contraction. How do we turn it on and how do we turn it off? So it's a sequence of events that leads to the action potential. And this is why it's so important to think about the different locations on the muscle where the action potential is coming in on the muscle surface or the sarcolemma, and where the cross-bridge cycle is happening in the sarcoplasmic cell. So it's going to be dependent on the neuron, right? The motor neuron innervating that muscle fiber. It's also going to be dependent on calcium being released from its storage area in the sarcoplasmic reticulum. And it's going to be a sequence of events, right? A cascade of events from the time that end plate potential. Remember, the end plate potential is the special type of action potential that we see on muscle when it receives that, that innervation on the muscle membrane. So from the time we have the end plate potential to the time that calcium is released and it's binding to troponin, that period, that cascade of events is what we refer to as the excitation contraction coupling. So here we want to think about the role of the neuromuscular junction. We want to think about the motor neurons that are innervating several muscle fibers or muscle cells. That collection is referred to as a motor unit, right? One motor neuron and all of the fibers it innervates is referred to as a motor unit. And then we also want to think about acetylcholine being released here. Remember, acetylcholine is the only neurotransmitter that is released in muscle, whereas we talked about norepinephrine in the autonomic nervous system, in the somatic nervous system, and with skeletal muscle, acetylcholine is the only neurotransmitter that is involved. We also want to think about the structure here, the motor end plate. So the motor end plate is the end of the muscle membrane, really. It's where we have a highly folded muscle membrane, and this allows for that excitation to spread. So it kind of increases the surface area a little bit to give that excitation the opportunity to spread. It has a high density of acetylcholine receptors, specifically ionotropic receptors. Remember, acetylcholine has two types, or binds to rather, acetylcholine binds to two types of cholinergic receptors. They can be nicotinic, which are ionotropic, or muscarinic, which are metabotropic. So the ionotropic receptors, the nicotinic receptors, are the only type of receptors that we see on skeletal muscle. So just to recap all of that from what we discussed in the somatic nervous system. And so here now, the motor neuron is going to generate the end plate potential on the surface of the muscle, which is referred to as the motor end plate. Now, what happens after that? After that, calcium gets released from the sarcoplasmic reticulum. And the role of calcium we just reviewed. Calcium is going to bind to troponin. It's going to uncover the binding sites by dragging tropomyosin away from those binding sites that are covering up those spots on the actin filament. And so if we have no calcium, we have no actin-myosin interaction. Therefore, we have no cross-bridge cycling. Therefore, the muscle will be in its relaxed state. In other words, we will turn off muscle contraction. If we do have calcium present, and that is going to be the effect of the end plate potential. So the end plate potential results in calcium release. And now if we do have calcium present, it binds to troponin, it drags tropomyosin off of those binding sites. It allows for that actin myosin interaction, and then it drags that actin towards the center of the sarcomere and basically allows for cross-bridge cycling and allows the muscle to shorten or to contract. So it's extremely important to think about all of these moving parts, and specifically how the end plate potential results in calcium release and how calcium release turns muscle contraction off. And the opposite of that, how the absence of calcium calcium release turns muscle contraction on. I think I misspoke there. And the absence of calcium release turns the muscle contraction off. All right. Okay. All right, so we can think about the steps of excitation, contraction, coupling, and let's just review those with the illustration here. So step one, acetylcholine is released from the axon terminal of the motor neuron, which is binding to the muscle fiber. Here's the muscle fiber down here. Here is the specific area on the muscle fiber referred to as the motor end plate. Notice how it is highly folded to increase the surface area. And notice how it has an abundance of acetylcholine receptors, specifically nicotinic cholinergic receptors. That acetylcholine crosses the synaptic cleft, binds onto these acetylcholine receptors, and generates the end plate potential, which is the specialized action potential that happens on muscle. That action potential is going to travel throughout the muscle. It's going to use these tunnels. These tunnels are referred to as T-tubules, and that allows this excitation, right, this end plate potential to travel deep within the muscle very quickly, okay? Instead of just moving on the surface, it's going to get deep into the depth of the muscle fiber. Remember, the muscle fiber has many muscle fibrils inside. So each of those needs to get this end plate potential, needs to get this excitation in order to result in the calcium release that will generate the phosphorus cycling. So that happens, and then that results in calcium being released from the SR. So here's the SR. And remember, we talked about this triad structure. The triad structure was formed by the T-tubule in the middle and the sarcoplasmic reticulum on either end, what we refer to as the lateral sacs or the lateral cisterns or cisternae. So these three structures together form what's referred to as a triad. And the reason that these structures have to be so close together is because the action potential has to get to the SR. It has to stimulate or signal the SR to go ahead and dump that calcium out so that it can circulate or float around inside the sarcoplasm itself and not inside the SR where it's just being stored. Okay, so the purpose of the T-tubule, allow the end plate potential to travel throughout the muscle and to stimulate the neighboring SR to dump the calcium out so that it can go ahead and engage with troponin, drag that troponin complex off of, or drag that, bind to that troponin, excuse me, and allow the troponin to drag the tropomyosin from the binding sites, exposing those binding sites such that myosin can interact with those binding sites, okay? So here myosin is, it is now in its high attraction form. It will bind to that binding site, do its dragging or its swiveling, its power stroke. And then in order for this process to cease, we have to get rid of that calcium. This is where it's so important to think about calcium turning things on, but the absence of calcium is needed to turn things off. So if we never put that calcium back into the SR, the muscle would just contract repeatedly without end, right? So in order for us to cease muscle contraction, we're going to have to pump this calcium back into the sarcoplasmic reticulum where it's being stored. We do that by way of calcium pumps, and then we're going to go ahead, cover back up those binding sites, and then myosin will no longer be able to engage with the